Hi, my name is Luke Reynolds, and my project is titled Identification of Amino Acid Residues Required for Dimerization of the Transferrin Receptor During Ferroptosis. Okay, so first of all, I should apologize. You can't see my entire board here because of the way this video is set up, but unfortunately this was the best way I could do it. So that's a lot of big words in my title, so I'm going to go through and break it down as I explain my project. So I'm actually going to start with the last word, ferroptosis. So ferroptosis is a form of cell death, much like the more commonly known apoptosis. Stemming from the root words ferrum and ptosis, which respectively mean iron and falling, it's no surprise that ferroptosis refers to cell death due to a lack of iron or too much iron within a cell. And ferroptosis is the first of three major facets of my project. The second is ECM attachment. ECM, which stands for the extracellular matrix, is a system of proteins and various different molecules that run through your body and connect all of your bodily cells to each other. As I've demonstrated in a few of my prior projects, detachment from the extracellular matrix, which typically causes cells to die, can allow cancer cells to metastasize. And this allows them to develop very resistant mechanisms which allow them to evade different types of cell death. So my project will focus on a few of the mechanisms that allow cells that have detached from the extracellular matrix to evade ferroptosis. The third major facet of my project is another word that was in the title, transferrin. So transferrin is a protein that is naturally found in all human cells that regulates or has a role in regulating iron intake within a cell. Of course, this is connected to ferroptosis because both are involving iron, and the goal of my project was to analyze and find out how I can manipulate that connection in order to cause cancerous cells, specifically 7860 and MDA-MB231, to die by ferroptosis. So I hypothesized that if I were able to use site-directed mutagenesis to remove 4 different cysteine amino acids within the transferrin protein, I would be able to prevent the creation of transferrin receptor dimers, which is 2 identical molecules bonded together, sort of like a monomer. I would be able to prevent the formation of the transferrin receptor dimers, which would allow cells to have a lack of regulation for iron and die by ferroptosis. Important distinction to make here, I sort of brushed over it, but the transferrin protein does not regulate iron on itself. The transferrin receptor protein does. The transferrin receptor protein is found in dimers, which are created by the transferrin protein. So my goal was to manipulate the original type A transferrin protein in order to prevent the formation of transferrin receptor proteins. So as I mentioned, I had 2 major steps in my project. The first step was site-directed mutagenesis, which I would be happy to share this procedure with you in depth, but for the sake of time, I'm going to just describe what it does rather than just how I did it. Essentially, it allowed me to go in and manipulate the proteins within the cells and remove certain amino acids and replace them with other amino acids or cut them out or add to them. In my case, I was conducting replacement mutations, which, as I mentioned, allowed me to replace 4 different cysteine amino acids with serine amino acids. So the reason I chose to replace cysteines was because cysteines have a chemical composition that allow them to, as has been demonstrated in many other proteins, that allow them to create disulfide bonds, which is the form of bond that is typically found in transferrin receptor proteins. I hypothesized that if I replace them with serine, which has a very similar chemical composition without the disulfide bond formation capability, it would prevent the formation of transferrin receptors almost entirely and allow cells to die by ferroptosis. Spoiler alert, my hypothesis proved correct. So I know it's hard to see this data, so I'll explain it as I go through it. But basically, through the use of Western blot images, I was able to identify 4 cysteines, cysteine 62, 67, 89, and 98, as being the cause of the transferrin receptor formation, which can be used in future research to further understand ferroptosis and hopefully one day create chemotherapies that will be able to save lives. Thank you.